Talking about meiosis again. Your warm-up today, number 2, was identify the ways and then explain how sex cells are different from the other cells in your body. So we're going to be comparing it back to mitosis. So if we're thinking about mitosis and the cell cycle, your sex cells go through the exact same stages that those things went through. So let's talk about the whole cell cycle, not just the little M phase, that was prophase, metaphase, anaphase, telophase. What is the name of the part of the cell cycle that is not the M stage? It's got one name. Interphase, right? So we'll start there. And if you remember, interphase in mitosis is divided up into different sections. Max, you remember the... Yeah. So do you remember the name of the different sections that interphase is divided up into, Max?
G1, S, G2.
Yeah, so the first three, G1, S, and G2. The M stage is the mitosis stage, so that's not part of it. But those are the three things, and these things, your sex cells, go through the exact same thing. The only difference is, it's called interphase 1 here because of the way it is. So it is similar to interphase in mitosis, right? Which means that if you remember, it grows in the G1 phase, it grows in size. In the S, and again, if you remember this, you certainly don't need to write it down a second time. In the S phase, that's when the chromosomes replicate, so it doubles the DNA that's inside of there. And then this says that each of the chromosomes consists of 2 identical chromatids that are attached at their centromeres, right? And then also happening in interphase is those centrioles go to opposite, or form, and they go to opposite ends of the cell. So that's just kind of the interphase part of this. Remember, G1, S, and G2. Those are the things. And they happen in meiosis just like they happen in mitosis. It's an interphase, so if you can remember what happens, you're doing all right. But in this stage, if you remember you can see the nucleus is what we're talking about, or the nucleolus inside. It's got a little dark spot in the middle. So it becomes visible, right? In interphase, that's when you can see the dark purple in the middle. The cell doesn't have stuff condensed into chromosomes yet. This is mitosis. It just happens twice. It's divided up into 2. There's meiosis 1, and then there's meiosis 2. And we're going to talk about a portion of meiosis 1 today. So what we're talking about is that normal cell division, right? It reduces the number of chromosomes by a half. If you remember, because the DNA doubles, so it's got twice as much as it's supposed to, and then when cell division happens, each cell gets half of what's there, so each cell gets a full complement of chromosomes. So in this stage of meiosis, or the DNA, the number of DNA gets cut in half to the appropriate amount for a regular cell. The difference is it doesn't stop here, because meiosis continues, right? It needs to continue until those cells have half of the DNA that they need to have. So it's divided up into 4 phases, and they're the same exact phases that mitosis has. So if you already remember those, you don't have to write them down. Again, I'm not going to make you remember the name of the stages, just like I didn't make you remember the name of these on the last test. But if it helps you to think about what's happening in each stage and knowing these names, then that's fine. But there will definitely not be a question on the test of what are the 4 names of the stages. Maximus? Right. Exactly the same. And the only reason that little 1 is on the end is because it's going to go through them a second time. So there's prophase 2, metaphase 2. So that's why there's a 1 on those. Today it's called prophase 1, the first step that happens. It is the longest and it is the most complex. And it is different than the stage that happens in mitosis in a couple of ways. This stage in meiosis takes about 90% of the time, right? So almost all the time is spent in this stage because there's a lot of stuff that happens in addition to what happens in the mitosis stages. So we're going to focus on the events that happen here, and these things you are going to be responsible for. So in this stage, the chromosomes become visible. So if you remember when we looked at these things under the microscope, in prophase is where you could start to see the nucleus break down and you start to see chromosomes inside of the nucleus. This thing called synapsis occurs which is where the homologous chromosomes come together to form a tetrad. And we'll pause there for a second so I can explain that to you. And it says right here, tetrad is 2 chromosomes or 4 chromatids. Sister chromatids, not chromosomes. So it kind of explains it to you. So if you've got this cell, and there's the cell, and inside of that, I'm going to draw a really big nucleus because we're going to need lots of room. So inside of there, right inside the dotted line, is the nucleus. And inside of there, you should recognize our chromosomes, right? At this point in time, chromosomes have formed. So I'm just going to draw the chromosomes as like X's around here, and we're just going to use that many of them. So we've got 4 chromosomes inside of there. So far, so good with my picture? Okay, this process, so this is like a verb. This is a thing that happens. What happens is, and this is different than mitosis, because if you remember in mitosis, once they condense, they just kind of line up along the center of the cell and on the metaphase plate in order to be pulled apart. What happens first here, though, is that these things actually come together and start to overlap like that. And they overlap with each other, and there's a reason for it. And when they are paired up like this unit right here is called a tetrad. Right? Because tetra means 4, I think. What, Shayla? Oh, I thought I lost you. And this is the beginning of the process that explains why you and your siblings don't look exactly the same. This is where that starts. If this didn't happen, just about every sex cell that would be produced would be the same as every other sex cell that's produced. So this is where they start to jumble up the DNA that's inside of them in this. But it starts with this process there. The homologous chromosomes, which remember a homologous chromosome is, right, that's the chromosome you might get from your dad. Here's the chromosome that you get from your mom. And together they're a homologous pair, or homologous chromosomes. They're the same chromosome. Even though I'm terrible at drawing, they should be the same chromosome, have the same kind of DNA on them. So these 2 chromosomes, the one you get from mom and the one you get from dad, pair up. Do you remember that we've got 23 sets of chromosomes in you? Right? So it finds its chromosome that it's a match to, and it pairs up. And when they do that, it's called a tetrad. Chromosomes that have like the same genetic information on them. So this one might have the code from your dad for your hair color, and this one has the code from your mom for your hair color. So they've got the same code on them, but this is the code your mom gives you for these traits, and this is the code that your dad gives you for these traits. What happens in meiosis, right, is first, these things are separated so that this becomes its own cell. This is what would happen if this doesn't happen. And this becomes its own cell, and then these 2 things divide up into 4. So you'd get, half of it here in that cell, and you'd get half of it here in that cell, and the same thing over here. Right? So you'd have a 50% chance of getting like exactly this part of your dad's DNA, or this part of your dad's DNA, or this part or this part. But that's not the way it works. What happens is, before it goes through that process to become sex cells, it actually goes through like a shuffling. And in this shuffling process, I'm just skipping through here. This is my version of it on here. This occurs, crossing over. And this is one of the big things that occurs in meiosis that's different than in mitosis. So crossing over, here's what happens. You've got identical segments of DNA on both, right? Like you've got the segment of DNA that codes for hair color or codes for eye color. And what happens is a piece of your mom's chromosome will swap places with a piece of your dad's chromosome. And it's totally random. So they shuffle your parents' DNA together and combine that to make chromosomes that are part mom and part dad, if that makes sense. So they shuffle them together. And there are so many different combinations that they can use because your DNA is so big that it's virtually impossible for a sibling to be identical to another sibling. Because think of all the genes you've got in your body that control you, right? For example, so this is dad's chromosome, this is mom's chromosome, And what we're going to do is I'll show you in like a bigger scale how this like swapping takes place. But as I do this, you should recognize that on these chromosomes, there are literally like millions of genes that are going to trade place. So we're going to do it with chunks instead of genes. But what happens in this process when they're joined here is your dad's chromosome, and we're going to take this top lobe or so, a part of it switches places with that. So it takes its part and sticks it there, and the mom's one joins up with the dad's side over here. So a piece of the chromosomes just swaps places. So now, when this thing divides up into a sex cell, you're giving your kids like part of your mom's DNA and part of your dad's DNA. Does that make better sense now that you can see it drawn out? And this I did it in one chunk, but this happens millions of times per chromosome, and you've got 23 chromosomes, so that's why it's very unlikely that you're going to look like a sibling, because for each chromosome this happens. Thousands, millions of little switches take place, and as a result, like you could have a sibling that's like 6'6", and you could have a sibling that's like 5'5". Just because you come from a tall family doesn't mean you're going to be tall. And this is why, because stuff gets switched up. Things trade place. It'd be like shuffling a deck of cards, essentially, is what's happening here to these two things. They get reorganized, little pieces trade back and forth in between them, and then the process of meiosis continues after that. So I tried to put it on a picture here as best I could. Right, if you've got the homologous pairs here, so here's like mom's chromosome and here's dad's chromosome, they come together and they swap places. Parts of them like go back and forth. This part of dad's goes over to mom's, that part of mom's goes over to dad's. And it happens not just in big chunks like that, but in little itty-bitty genes all the way across the thing. That those things are trading places. And that's how you get this word diversity amongst offspring. You get differences between offspring. Like you could have a person in your family with blonde hair and somebody else would have brown hair. And that's how it can happen. That's the idea behind it. And that's where all that variation comes from. So that is the end of the notes that I wanted to get through yesterday.